Okay, to begin, please open the Audition Instructor PowerPoint. Find the slide that says Begin Here. I will give you a moment. All right, let's advance slide. What you're looking at here is an image that we ended class on Friday that shows how sound waves are transmitted through the ear apparatus all the way into the cochlea. The air waves strike the eardrum, moving the eardrum. The eardrum moves the auditory ossicles. We know them as hammer, anvil, and stirrup. As the ossicles move, they press against the oval window of the cochlea, setting up movements in the fluid inside the cochlea. You can see that fluid is in something called the vestibular canal. The vestibular canal continues the length of the cochlea and actually communicates or connects to another canal at the bottom or the outside. Between those lies something called the basilar membrane. And if you look right now, find basilar membrane, you'll see it says vibrations of this membrane stimulates the sensory hair cells in the organ of Corti. Hair cells are going to be what we focus on. So let's advance slide. Here's a close-up illustration. You can see the vestibular canal. That is where the waves start from the oval window down the vestibular canal. They then connect at the very tip of the cochlea and the wave comes back down the tympanic canal. Between those two, you can see the organ of Corti, which we will simply refer to as the basilar membrane. So if you think about it, if there's a wave passing from the vestibular down through the tympanic, that wave is going to wiggle the the basilar membrane. As the basilar membrane goes up and down, interesting things happen. Next slide. Here's a close-up of the organ of Corti, or basilar membrane. And you can see, starting on the far right, the outer hair cells. So these outer hair cells are bent as the basilar membrane moves up and down. If the basilar membrane moves up and down a lot, then the inner hair cells are also bent. It's when the hair cell is bent that exciting things start to happen. Next slide. Here's another illustration of that same thing. Basilar membrane, outer hair cells, inner hair cells. So as the basilar membrane moves up and down, the hairs, which in this illustration are called stereocilia, we will simply call them hair cells, bend because they're rubbed against the tectorial or the top of the basilar membrane with kind of a shearing force. It bends them. They're going to set up EPSPs in the first synapse, which you can see right there in yellow beneath the outer hair cells. Those EPSPs generate action potentials which pass out the auditory nerve in the lower left of this illustration headed to the brain. Next slide. Now, here's what's important to understand. You can see in the top left the cochlea illustrated as if it were being rolled out flat. So if you take that snail-shaped cochlea and roll it out flat, here's what we know. The hair cells at the very top, or very beginning, I should say, the very beginning, the base, respond to 20,000 hertz. That's the top of the human hearing sensitivity, the top pitch. So those hair cells are only going to respond to 20,000 hertz. At the very apex, the very tip, you can see the hair cells are responding at very low pitch sounds, very slow vibrations, 100 hertz. And you can see in the illustration to the right why that is true. The narrow, thick base is tuned for high frequencies. The wide, thin apex is tuned for low frequencies. Now, why is that the case? Because the basilar membrane resonates. Now, what is resonance? Resonance is where sound vibrations, when they strike an object, that would vibrate if it were activated at the same frequency as the sound wave striking it. So that object will begin to respond to the sound wave by vibrating. Let me say that again. If a sound wave hits an object, and that object would vibrate at the same frequency as the sound wave striking it, the object begins to vibrate or resonate. The resonance in the basilar membrane depends upon thickness and tension. So, the narrow, thick base resonates with a very high frequency. The wide, thin apex resonates with a low frequency. Or in other words, when a sound wave comes into the cochlea, some single location along the basilar membrane will begin to resonate or move in response to that wave. For example, the middle of the cochlea, if you look up at the upper left illustration, the middle will resonate or vibrate to a sound of about 2,500, between 2,000 and 3,000 hertz. Can you see that? Look for the 1,000, look for the 4,000, right between them, between 2,500 and 3,000 hertz. That is where the basilar membrane resonates if a sound of 25 hertz comes into the ear. Thus, each location on the basilar membrane is tuned to resonate to one frequency. Only the hair cells in the location that's resonating would be bent. Thus, we know frequency of sound, or the pitch of the sound, based on which hair cells are responding. This is what is known as the place theory of pitch perception. The place on the basilar membrane that is resonating causes the hair cells in that place to respond. According to the law of specific nerve energies, if the signal comes into the brain from a place that corresponds to, let's say, 3,000 hertz, we would perceive 3,000 hertz. Next slide. Here is another illustration that just shows you the same thing. So if you look at the left side of the image, you'll see there's the base of the cochlea, the big wide open pipe, that is the canal into which the wave is first introduced by the movement of the ossicle. The wave travels down that canal, the vestibular canal, all the way to the tip. You can see the apex. It passes around the tip, travels back down the tympanic canal, the smaller canal, and as the wave moves around the basilar membrane, it sets up movement. So you can see in the panels to the right, if the wave is very high frequency, the basilar membrane at the base moves. If it's very low frequency, the base at the basilar membrane at the apex moves. Now, here's the problem with this model. Next slide. If the place that corresponds to, let's say, 3,000 hertz is moving, well, the basilar membrane attached to that place on either side is obviously going to move as well. The basilar membrane is, after all, one continuous membrane. So that means that the hair cells for 3,000 would respond, but because the adjoining areas, the part of the basilar membrane either side would also move, the hair cells for 2,900 and 3,100 would also respond. Perhaps a little less, but they'd respond. The same for 2,800 and 3,200 and so forth. Thus, the question becomes, how do we get precise pitch perception? Or how do the auditory receptors fine-tune the perception of pitch?